Let's go over the parts of the brain involved with memory. There are two memory circuits, a medial circuit involving the hippocampus and a lateral circuit involving the amygdala. The hippocampus is critical for episodic memory, whereas the amygdala is critical for emotional aspects of memory. Functional impairment in both circuits is necessary for dense amnesia, but less severe memory impairment can also happen from lesions that only affect one of the two circuits. I'm going to teach you these two complicated circuits later using an easy story about a hippo and some other memory tools involving how the amygdala is so emotional. Before we talk about the two circuits, let's back up and talk about some important parts of the brain. The hippocampus is part of the limbic system and plays a crucial role in the formation of new memories, particularly episodic memories, meaning memories of specific events or experiences. The hippocampus is involved in the initial encoding of information, as well as spatial memory and navigation. You can remember this by thinking about hippos just traveling along down their little paths in Africa. They never get lost, they know exactly where they're going. The hippocampus contains the dentate gyrus, the sectors of Ammons horn, and the subiculum. How do we remember this with our hippo story? Well, the hippo has those huge little tusk things coming out of their mouth, or huge teeth, so they need to go to the dentist a lot. In fact, their little teeth are almost so big that they're more like a horn. Let's call them horns, I think. And have you ever seen a hippo run underwater? This is a real thing. They run so fast, they're like submarines. The dentate gyrus is important for pattern separation, spatial learning, spatial navigation, and temporal association. In other words, sequencing the order of events or organizing and retrieving memories in a coherent order. Our hippo knows exactly how to get to the dentist, right? Hippos are good with navigation. They're also good at picking up on patterns. When our hippo goes to the dentist, he expects a certain sequence of events. First, he's going to meet with the hygienist, then he's going to meet with the dentist, then they're going to give him his little goodie bag, then he's going to navigate home. Ammons horn consists of several distinct regions, including the cornu-ammonous regions, or CA regions, which are numbered CA1, CA2, and CA3. Each section of Ammons horn has specific functions and characteristics. So our hippo has three different types of horns here, as you can see. Alright, don't look too closely, but just trust me, he has three different types of horns. CA1 is spatial, CA2 is social, and CA3 is associative memory and pattern recognition. What do I mean by this? The CA1 region is the spatial region. It is heavily involved in the formation and consolidation of declarative memories, particularly episodic and spatial memories. It processes information related to spatial navigation and contextual learning. CA1 neurons contribute to pattern separation, or the process of distinguishing between similar experiences or events and forming distinct memory representations. This function is critical for forming unique memories and avoiding interference between closely related memories. The CA2 region is the social region. It has been implicated in social memory and recognition of members of the same species. It plays a role in encoding and retrieving memories related to social interactions, social hierarchy, and recognition of familiar individuals. In addition, CA2 neurons are uniquely sensitive to the neuropeptide vasopressin, which modulates social behaviors and social memory. Vasopressin receptors in the CA2 region contribute to the regulation of social behavior and aggression. The CA3 region is important for associative memory and picking up on patterns. It is involved in pattern completion, the process by which incomplete or partial cues trigger the retrieval of complete memory representations. It plays a role in recalling memories based on partial or degraded cues and reconstructing the entire memory trace. Notably, the CA3 region exhibits a higher rate of adult neurogenesis compared to the other hippocampal regions. The subiculum is situated between the hippocampus and the entorhinal cortex and serves as a critical interface between the hippocampus and the broader cortical and subcortical regions. It is involved with spatial navigation, memory consolidation, emotional processing, decision making, reward processing, temporal processing, and neuroplasticity. Our hippo navigates underwater like a submarine. He remembers where to go because those memories of underwater routes are consolidated. Sometimes he gets emotional because he thinks he's too fat and he has to make tough decisions about what to eat. He has to decide if the reward of eating it is worth it. He remembers the last time he ate a huge watermelon and then there was this whole sequence of events that was terrible, so he might decide not to eat it. In addition to the hippocampus and its three parts, you should also know about the parahippocampal regions, which are other structures within the medial temporal lobe that are important to memory. These include the perirhinal cortex, parahippocampal cortex, and the entorhinal cortex. The perirhinal and parahippocampal cortices appear to receive input from different cortical regions. The perirhinal cortex receives more of the non-spatial or what information from the anterior temporal lobe. Perirhinal, like rhino. What is that, a rhino or a hippo? Whereas the parahippocampal cortex receives more posterior, medial, spatial, or where information. Posterior to all of this, or toward the back of all of this, the entorhinal cortex receives the input from the parahippocampal and perirhinal cortices. The lateral entorhinal cortex is part of the ventral spatial stream, and the medial entorhinal cortex is part of the dorsal or non-spatial stream. The amygdala is another limbic system structure located near the hippocampus. It is involved in the processing and consolidation of emotional memories. The basal forebrain consists of the septal area, the diagonal band of Broca, nucleus accumbens, septi, olfactory tubercle, substantia innominati, which contains the nucleus basalis of Maynard, the bed nucleus of the stria terminalis, and the preoptic area. The memory functions of the septal nuclei and the nucleus accumbens are associated with reward and reinforcement. The diagonal band of Broca and substantia innominati are both associated with acetylcholine, which promotes learning and memory consolidation by enhancing synaptic plasticity and facilitating long-term potentiation. The olfactory tubercle is important for processing emotionally salient olfactory stimuli and modulating emotional responses. The bed nucleus of stria terminalis is involved in processing emotions, stress, social behavior, sexual behavior, reward, and autonomic functions. The preoptic area is involved in regulating diverse physiological processes, including thermoregulation or temperature regulation, sleep-wake cycles, reproductive function, endocrine function, fluid balance, and social behaviors. Moving on from the basal forebrain to the medial diencephalon, which includes the thalamus, hypothalamus, and epithalamus. Specific nuclei within the thalamus, such as the dorsomedial nucleus, have been implicated in memory consolidation and retrieval processes. Internal medullary lamina, IML, are myelinated fiber tracts that separate the ventral dorsal and anterior posterior planes. The IML contains fibers relevant to memory from the mammillothalamic tract, which travels to the anterior thalamic nuclei, and the ventral amygdalofugal pathway, which travels to the dorsomedial thalamic nuclei. Thalamic amnesia is most associated with lesions affecting the IML and mammillothalamic tract, not the dorsolateral thalamic lesions, which was previously thought. This is where we turn to the two circuits. The hippocampally-based medial limbic circuit involves a projection from the mammillary bodies to the anterior thalamic nucleus and subsequent thalamic projections to the cingulate gyrus and cingulate projections via the cingulate bundle, which extends back to the hippocampus. The amygdala-based lateral limbic circuit projects from the amygdala to the dorsomedial nucleus of the thalamus, to the orbitofrontal cortex, to the uncinate fasciculus, to the amygdala. Okay, don't freak out. I'm going to help you remember this. Remember that the medial circuit is for episodic memory, meaning memories of specific events or experiences. So it makes sense to remember it with a story. Once upon a time, there was a hippopotamus. Mammals like him have big bodies. They get that way by using a fork to eat a lot. In this story, the hippo travels along a pathway with all the other mammals until he gets to the anteater. The anterior thalamus. Then he sings a song to all the other hippos. The lateral circuit is for emotional memory, obviously, because it starts with the amygdala. Sometimes when you're super emotional, you feel like you're in some kind of fugue state, just traveling along in this state of fugue. That is, until you open the door to the thalamus, the sensory relay station that brings you back to your senses. As you return to your senses, you see an orb in front of you, and you think it must be the spirit of your uncle. So then you start to cry again because you miss your uncle, which is that amygdala acting up again. Please like this video and subscribe if you want more free study videos for neuropsychology board certification.